Then I pass over the session to the organizing committee. Then, thank you.
Thank you once again, Dr. Rosita, and also to our three presenters for your useful sharing on the current status and research being undertaken at your respective countries. So also to the participants, keep those questions coming on the chat box. The next session is going to be chaired by Mr. Yaakob Ahmad. Mr. Yaakob is at present the advisor to the International Tropical Fruits Network. He possesses vast experience and knowledge spanning more than 4 decades in the horticultural field, particularly on various aspects of production and activities along the value chain of horticultural crops. So without further ado, Mr. Yaakob Ahmad, the floor is all yours.
Thank you, my favorite colleague. Anyway, okay, for this session, we'll have 2 speakers. But before this, I just like to remind participants that we'll be having a panel discussion after this. So I would like all speakers to be on board, because it'll be a very short one, but I just need to get some feedback to you. Okay, for this section, we have 2 speakers. One is Dr. Loganathan from India, and the other one is Dr. G. Loma, I hope I got your name right, from Belgium, based in Uganda. So we will start with Dr. Morgan Loganathan. Dr. Morgan is the principal scientist in plant pathology, Division of Crop Protection, ICAR, National Research Center for Banana, Trichy, in Tamil Nadu, India, right? And Dr. Loga, he has a lot of experience. He started with vegetables, cashew, and now he's working with bananas. Dr. Loga has also received many professional recognition, awards, prizes, certificates, fellowships, and he has also published more than 60 papers. Dr. Loga, we will teach for us this evening is bacterial diseases of banana in India, overview on status, characterization of pathogen and management practices. Dr. Yoga, if you're online, okay, the space is yours. Thank you very much. Your line is off. Yeah, okay.
Thank you very much. Should I need to share my slide or already which I sent you?
Well, please, if you are comfortable with sharing, I think it's better for you to share your slide. Yeah.
Okay.
Thank you.
I'll check. Sorry, I'll check it. Okay. Okay, good afternoon, everybody. My topic is on bacterial disease of banana in India.
Can you put yours on full screen, please?
Yeah, I have put my slide in full screen.
Yes, but it's not.
Or could you please share my slide which I sent today early morning?
Okay. Then we should. Okay.
Yeah, something problem because
okay.
It's showing full screen for me.
Okay, we will do it for you. Chris, can you please put Dr. Loga's sign on, please? Thank you.
Yeah. Now it is okay.
Yeah. Okay. Is that right?
Yeah, it's fine.
It is okay now.
Thank you.
Yeah. Today's my topic is going to be on status, characterization of pathogen management practice followed in India, all about bacterial disease of banana. Can I have next slide, please? You know, India is a world leader in banana, okay. Which has 0.86 million hectare area of banana, which tend to be 30 million tons of production every year. It hosts diversified banana across the country. Different varieties are being grown. Can I have next slide, please? Major production hurdles are biotic stresses, start from fusarium wilt, leaf spot, viral diseases, bacterial disease. In viral disease, bungee top, streak, and cucumber mosaic virus are major one. So today, what I'm going to cover is bacterial disease status and characterization management. Then I have next slide. So initially, in the year 2018 and 19, we have conducted a vast survey covering all over India, from southern part to northern part, east, west, and central part of India, in all those states like Tamil Nadu, Karnataka, Andhra, Maharashtra, Madhya Pradesh, and so on. So we have collected the information about bacterial diseases. Next slide, please. So this is a status report. Mucovility is not present, blood disease not present, Xanthomonas is not present, this fortunate thing. But there are few reports which earlier they reported raised to of Ralstonia solanacearum in West Bengal, but that's all. It's possibly not confirmed results because we do not have a recurrent problem in those areas, and we did not receive any report further. But rhizome rot is present all over in India, in across all commercial cultivar is a major problem in banana cultivation. Next slide, please. This is a status of rhizome rot in different banana growing area. In Tamil Nadu, it has a 3.9 to 30% incidence, both the sucker planted commercial varieties and also tissue culture planted one. But tissue culture planted seedlings had more incidence than the sucker planted. Next slide, please. This is a symptom of rhizome rot present in Naipuvan, which was taken from Tamil Nadu. Next slide, please. This is there are some associated infection of rhizome rot also along with the Fusarium wilt, because Fusarium wilt is possibly primary cause of the disease, but the secondary infection could be rhizome rot that is present in Naipuvan variety. Next slide, please. This is a severe incidence of rhizome rot in granite, the most commercially cultivated one in Tamil Nadu area. This incidence was about 30%. This is pisciculture grown garden. Next slide, please. This is a typical symptom of rhizome rot. If you look at the inner core region, that is showing rotting, what we call hot rot. And also another typical symptom is the primary shoots get affected by rhizome rot. The later on, after 2 to 3 months, the lateral shoots forms, but that will not come into uniform yielding when other plants get yielded. Next slide, please. This is a scenario of rhizome rot in other part of country, that is southern Andhra Pradesh, Karnataka, that is southern part, then eastern part, West Bengal, and different varieties. There also we could find 3 to 20% incidence across the cultivar, across the state. Next slide, please. This incidence we noticed in Karpura Chakrakali AAB group at Andhra Pradesh. Next slide, please. This status of rhizome rot in central and eastern and northern part of India. There, if you cross central part of India, then you could find only granine as a most commercially cultivated variety, but mostly they go for a tissue culture one. The incidence was heavily in the granine variety, around 6 to 20%, the maximum across the state in different part of country. Next slide, please. This is a summary status of rhizome rot of banana. Incidence is ranged from 20 to 30, that to the most commercially cultivated one, granine, had more incidence. Mostly this rhizome rot comes in early stage of establishment, 2 to 4 months after planting. Among the sucker planted varieties, the Naipuvan had more incidence than the Nandrin and granine. Then this rhizome rot also associated with Fusarium wilt in some cases. Next slide, please. This is about characterization of the pathogen. When we characterize the pathogen isolate collected from all over India, we could find mostly Lactobacterium curtovirus as a causal agent, which has been also reported by the several workers across the India. But there are 3 isolates out of 60 could not produce the unique colonies on crystal violet pectate medium, basically used for isolation of Lactobacterium curtovirus. So we were interested to know the 3 unique isolates, the characterization. So that samples collected from Naipuvan in Tamil Nadu, granine from Tamil Nadu again, Telachakaragali from Andhra Pradesh. So these 3 are the isolates, NPK, GTC, and 1-1B. These 3 isolates we specifically characterize further to know the nature of the pathogen. Next slide, please. So this pathogenicity test to be performed on granite. It showed incidence after 35 days of inoculation under pod culture condition. Next slide, please. The NAPU1 isolates be collected from a sucker planted NAPU1, then again be inoculated in NAPU1 variety suckers. So we could get incidence around 30 to 35 days of inoculation in all the inoculated plants. Next slide, please. So further, we characterize these three isolates biochemically by using different biochemical method. Start from gram staining with all the isolates were gram negative. Catalase positive, then catalase positive means it could be aerobic. So again, we tested for oxidase, it was negative. So that means that is not aerobic, it could be a facultative aerobic. So again, we tested with carbohydrate utilization. It was utilizing glucose, the maltose. Again, these were positive. So there are some reports which says that these two maltose and citrate specific for Klebsiella varicola, it could distinguish the Klebsiella pneumoniae. So our isolates possibly could be a Klebsiella varicola based on the positive results showed in maltose and citrate utilization. Next slide, please. Again, we further gone for molecular analysis through cystine sequencing, all clubbed together in the phylogenetic tree with the type stain of Klebsiella varicola and also the stain which has been reported from China. So here also we confirmed that these three isolates are Klebsiella varicola. Next slide, please. Again, we have gone for sequencing with RNA polymerase beta subunit sequencing. The results were concurrent with earlier results. So this again we confirm these isolates are Klebsiella varicola. Next slide, please. This is a summary of characterization. Crisply we could say besides Bacterium, Klebsiella varicola also caused the rhizome rot in India. Next slide, please. This report has been published in Plant Disease very recently in 2021. Next slide, please. So actually, based on our experience, this pathogenicity of rhizome rot, it takes a long time, about 30 to 35 days. We wanted to develop a quick pathogenicity test by fabricating soil heating facility by certain mechanisms. So by this method, we could get a pathogenicity result within 10 days. So all I would like to say that specific temperature is required for bacterial infection, especially rhizome rot. For Pectobacterium, it was around 30. For Klebsiella varicola, it was 35 to 40 degree. Next slide, please. This validated the result of the previous one. Next slide, please. So etiology, that's all about characterization, then pathogenicity test. Then coming to the etiology, based on the literature, we could know that the spread of disease is mainly through infected planting material. Then hot weather followed by high rainfall favors the disease. Then most of the cases, ill-drained soil had more the disease incidence. Then other factors like Fusarium wilt and comb borer favor the secondary infection of rhizome rot pathogen. Next slide, please. Next, come to the management practice. Normally, we recommend the use of healthy planting material, either it may be a sucker or TC. It is obvious one. Then in 2016, through all India network projects, we have found out two module worked well against rhizome rot management. First one is drenching of streptocycline, that is 500 ppm on second and fourth month after planting, and growing sunnem or cowpea in interspaces, mainly to grow these intercropies to reduce the temperature, soil temperature, and also increase the soil organic matter and rhizospheric microbiome. So in this up to 6 month intercropping, and after that it should be plowed into the soil. So that is the first module we recommended. Then second module is application of bleaching powder at the rate of 6 grams per plant, monthly interval up to 5 months. Then again trenching with 500 ppm retrocycling, 1 to 2 liters per plant a month after planting. Again, application of Trichoderma viridi at the rate of 50 gram on second and fourth month after planting. Then again, growing intercrop with cowpea or sungum. These two modules worked well for rhizome rot management. Besides that, bleaching powder alone also could work some extent against rhizome rot. Next slide, please. Unfortunately, these antibiotics had ill effects, like it developed pathogen developed resistance in agriculture system. So, Government of India in the year 2021 December 17 imposed a ban on import, manufacture and formulation of this antibiotics in India. It came into effect from February 1, 2022. So it is going to completely ban from 1st January 2024, because these antibiotics again used in human disease management, especially tuberculosis, which is very notorious disease in human. So based on this, government insisted us to find out the alternate chemicals. Next slide, please. So we have tested some copper-based fungicides which worked against the pathogen of rhizome rot. So copper hydroxide at the rate of 0.2% worked well against Pectobacterium curtovorum. Copper oxide chloride 0.2% was effective against K. varicola. These results are across the isolates, because copper hydroxide also worked against Klebsiella varicola, but certain isolate could not, and vice versa in case of copper oxide chloride. Next slide, please. We have also some biocontrol agents for management of this disease, because basically in India, 455 million tissue culture seedlings are produced annually and planted. So the problem is more in tissue culture plants, because these are all tiny in nature. It succumbs to the rhizome rot very easily. So primary thing is, what we can do is biopriming is a primary need of things for controlling disease in tissue culture plant. This has been done in our center, also elsewhere in State Agriculture University. That is in the pipeline for in future commercialization, because whatever the biopesticides or fungicide, whatever it may be, but it should be registered under CIBRC. That means Central Insecticide Board Registration Committee, Government of India, which is located in Hyderabad of India. So it should approve that chemical for use in agriculture, then only it can be commercialized legally. Next slide, please. So these are all some promising biocontrol agents we have identified against this rhizome rot. The previous earlier it was baseless or ariopectic. Again, previous Bacterial. This biocontrol agent worked against rhizome rot. Next slide, please. This is some of the plant growth promoting activity of this rhizobacteria tested in granite seedlings under glasshouse condition. Next slide, please. This is a biogarden plant ready for field planting. This year we are going to evaluate under field condition. Next slide, please. This is about the management practices what we follow in India. Besides that, since only rhizome rot is present, but other exotic disease, what we call Xanthomonas wilt or Buckdog or blood disease, also we have to sensitize the all stakeholders. We constantly conduct the training program to the research group, farmers group across the India. Next slide, please. Also, we publish the news in leading TV channels. We have given this information in the TV channel to sensitize the people about new bacterial disease, and also already existing one for management practice. Next slide, please. We also develop a mobile app for rhizome rot for other disease and pest also. So through which we also sensitize the presence of rhizome rot disease and management practice, and the new disease about Xanthomonas wilt, Mocha wilt, blood disease and Buckdog, how to identify if anything it's noticed at any part of country. They have to inform us through email or mobile number that has been placed in the mobile app. Next slide, please. So we also discussed through WhatsApp, through scientific grouping, and also the farmers group. We regularly interact with each other about the ongoing problems faced by banana stakeholders. Next slide, please. That's all about my presentation. This is a conclusion way forward. Our humble submission to the researcher is before reporting it as a new disease, that should be thoroughly analyzed through cultural, morphological, biochemical, molecular basis, and again it should be rechecked. Then only they can go for reporting to avoid the problem in future. So another thing is we can go for low volume, high shelf life bioformulation for effective management of rhizome rot. So it should be validated under multi-location trial. Again, it should be registered under CEBRC under Indian condition. So finally, thing is that we have to go for commercialization of the technology for benefit of the stakeholders. Next slide, please. So next part is management part is what we develop is that should be sensitive, that should be given to the stakeholders through awareness. So we can use the social media like WhatsApp, Facebook, and YouTube to sensitize and also to give the information about what the technology we develop at different centers. In future, we can also go for advanced technology like IoT. This is our of the need because by which through machine learning or deep learning, we can get the things in immediately and we can give the solution at the earliest. Then future of this thing is nano biopesticide, maybe a live cell. Live cell is not possible because it is a nano. Maybe the secondary metabolites or the product obtained from biocontrol agents can be converted into nano-based formulation so that it can be easily used for management of these diseases. Can I have next slide, please? This is our working team, our leader director, myself, Dr. Tangavilu, he is a senior colleague. Then down one is a technician, Mr. Muthuparadi, who is working under this project. Next slide, please. Finally, I would like to thank the funding and support given by our primary institute, ICAR, and also Institute National Research Center for Banana. Besides that, the different scientists from situated at different location of India who helped us in during survey. Dr. Bhagavan, Snehalada, Devpa, Das, and VK Misara that I missed, sorry. Then other scientific and technical staff from KVK. KVK is a robust system in India which gives the information directly to the farmers. They also helped us for collecting the information about the bacterial disease. Besides that, the private players also helped us. Dr. KP Patel, who helped during visit to the Maharashtra area. Next slide, please. Finally, I thank International Tropical Fruit Net for giving me the opportunity to share my presentation among the stakeholders. Thank you very much.
Thank you very much, Dr. Loganathan. Excellent presentation. I think what we do is we're really short of time, but I'm going to ask you a very short question. I noticed, like, okay, the situation in India for bacterial disease is a little different from Malaysia or in Indonesia or Philippines, because you speak more about the rhizome rot, yeah. And I think the causal agent is like Awinia. It used to be Awinia, right? It's bacteria. But the point is, I just want to. I know that most of the infection actually for the AAA, but the Cavendish type varieties, right?
Yes.
Most infections. So I just want to find out whether are they also common on the ABBs or AAB or on other varieties? Yeah. So that's number one. And number two is I'm a bit surprised, like for tissue culture, supposed to be clean plant materials, you still get infected. That means it's more of a soil problem then. So when it's a soil problem, it can also be other organisms. It can be FOC, it can be some other pathogens. Just my comment. Thank you.
The first question I would like to answer is most widely found out causal agent is Pectobacterium cartovorum. Earlier it was called Erwinia cartovorum. That's both are same. Then in few cases we could find Klebsiella vericula. Only 3 samples we could find. That is not widely occurring in India. Next question is this this is more prominent in commercial cultivar granine because granine is most widely used in central and northern part and east-west part of India. So mostly they go for tissue culture planting material. Basically, the tissue culture planting material will have the small in the comb size compared to the sucker plant, because sucker will have 15 to 20 centimeter size. So whenever the intercultural operation happen, injury happens to the seedling, the bacterium easily reaches the center point, it attacks the shoot. So the rotting happened obviously. So that is the main reason. Otherwise, we could not find any other alternate reasons found out.
Okay.
Thank you very much.
Thank you much. Okay, I think since we are a bit short time, we go ahead with Dr. Gee's paper, and then after that we'll just open it up. And finally, we'll spend maybe 5 to 10 minutes on panel discussion, and I hope all the speakers will be around. Just a little bit of interaction. We just want to get some kind of summary for this workshop for today.